Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is the ethylene signaling pathway. Okay, so before going to this topic, I want to say that if you are the newcomer, so please you just subscribe and sharing this channel. Okay, if this is helpful to you, and obviously just you hit the bell icon for getting all the notifications. Okay, and generally I upload my videos in Saturday, Sunday, and sometimes Tuesday and Thursday. So if you just after subscription hit the bell icon for getting all the notification when I upload my videos. Okay, so this is the ethylene signaling pathway, and here that ethylene signaling pathway, the receptors, ethylene first bind with the receptors, and the receptors, these have proper features. That means these are dimeric, multipass, transmembrane proteins and with copper containing ethylene binding domain. So what are the features? These are the dimeric. Dimeric means in the both side of the receptor ethylene can bind. Okay. Multipass means there are the passes. Pass means the ports, you can say, or the channel through which this can bind. This can pass. Any ethylene can pass. That means the receptor has the multi passes through which the ethylene can pass. Okay. And this containing the copper containing domain. Okay. There is important thing is the domain present. And they are grouped in a family of S members, ETR1. These are the examples some. ETR1, ETR2, the full form is that ethylene receptor 1 and 2, ERS2, that means the ethylene response sensor, and EIN4, that means the ethylene insensitive 4. These receptors are primarily localized in the ER lumen. Okay. ER lumen. So receptors were present. These receptors are the present in the ER lumen, and their these receptors are generally negative regulators. Negative regulators means two kinds of regulators are there. One is the positive regulators, another is the negative regulators. Positive regulators are those receptors where the hormone can bind, then the receptors become activated. Okay, and here the negative regulators means when the ethylene can bind with the receptors, this inactivates the receptors. Okay, inactivates the, when these receptors bind with the ethylene, it inactivates the receptors. So positive means when the receptors becomes activated, and negative means when the receptors become inactivated. That means when hormones bind with the receptors may be inactivated or may be activated. Okay, or might be activated or might be inactivated. If inactivated, that means the negative regulators. If activated, that means the positive regulators. Okay, and here the enzyme receptors have the non-cyclic domain. They have the non-cyclic domain which contains copper atom, and that binds the ethylene cytosolic ethylene. One second. Sorry, there was a mistake that there is the AND and must have. Okay. So what is saying that the receptors that have the non-cyclic domain, and these contains a copper atom. And what is the role of this copper atom? This binds ethylene and a cytosolic intercellular histidine kinase domain. Clear? So this bind ethylene and also histidine kinase domain. This is the role of the copper atom that present with the receptor. The two can bind with the copper atom. One is the ethylene and another is the histidine kinase domain. Now, when the ethylene bind with that receptor, the inhibiting histidine kinase domain and the downstream signaling pathway emanating from it. That means what is saying that when ethylene binds with the receptor, that inhibiting the kinase domain. Okay, that histidine kinase domain become inactivated or inhibited, and that's why downstream signaling pathway of the ethylene that is occurring. And in its unbound active state, receptor activates serine threonine kinase and CTR1. This CTR1 means constitutive triple response 1. The function of CTR1 that depends on the ethylene signaling pathway, in the ethinyl signaling pathway on its serine threonine kinase activity. Okay, and association of N-terminal domain with the ethylene receptor. So, if this is if in its unbound condition, that means when the ethylene cannot bind with the receptor, then the receptor remains in active state. Okay, receptor remains in active state, and this activates the serine threonine kinase. Okay, so when receptor becomes activated, it activates the serine threonine kinase and also CTR1. This CTR1 is what? This is the constitutive triple response 1. So both two are activated if the receptors becomes activated. Now, what is the function of this CTR1? The function of CTR1 that it helps in the signaling pathway. Okay, this helps in the signaling in the ethylene signaling pathway, and it depends, it's a function that depends on the serine threonine kinase activity. Okay, serine threonine kinase activity, and generally CTR1, that the function of the CTR1 that inactivates the genes responsive, ethylene responsive genes inactivated by the activation of the CTR1. This is the function of the CTR1. Okay, and its function also depends on the association of the N-terminal domain with the ethylene receptor. Okay, so two important things are, or two important factors are involved in the activation or inactivation of the CTR1. One is the serine threonine kinase activity, and another is the association of N-terminal domain with the ethylene receptors. Two important factors are very much involved in this CTR1's functions. Okay, and the active CTR1 that stimulates the ubiquitination and the degradation of the nuclear gene regulatory proteins, so called the EIN3, and which is required for the transcription of the ethylene responsive genes, and ethylene binding inactivates receptors alternating. Okay, this is the another thing. So what is saying that this active CTR1, when the CTR1 becomes activated, it stimulates the ubiquitination and the degradation of the regulatory proteins. Okay, gene regulatory proteins, and this is known as the EIN3. Okay, EIN3, or you can say that ethylene insensitive. Okay, insensitive 3, which is, what is the role of this EIN3? This is important for the transcription of the ethylene responsive genes. Clear? This is important way. This is important for the transcription of the ethylene responsive gene. So these are the flowcharts where the ethylene absent, that means the activated histidine kinase domain. Here the histidine kinase domain that becomes activated. So CTR1, it is activated here also, and CTR1 if activated, that the transcription factor, this through this EIN3, polyubiquitination and the proteasomal degradation is occurring, and that's why the ethylene responsive genes that off. Clear? But when the ethylene present here, that means the histidine kinase domain that becomes inactivated, and if this is inactivated, so CTR1 is also inactive, and if CTR1 is also inactive, then EIN3 that helps in the activation of ethylene responsive genes. So transcription is occurring. Clear? So if CTR1, its role is very important. CTR1s when active, CTR1s when actives, okay, that means the EIN3 that helps in the ubiquitination and the proteasomal degradation. Both the same proteins, but the activation of the CTR1 and the inactivation of the CTR1 that controls the function of the EIN3. Clear? If CTR1 is activated, that means its role is the ubiquitination and the proteasomal degradation. That means of the factors, transcriptional factors. So that's why the genes that remain off, ethylene responsive genes that remain off. But if CTR1 is inactive, then its role, EIN3's role is very important for the ethylene responsive genes activation. Clear? And here another important thing is that the ethylene binding inactivates the receptors. How? It just changes the conformation of the receptors for further binding with the membrane or membrane. That means ethylene binding inactivates the receptors, alternating the conformation so they no longer bind with the CTR1 and CTR. Okay, no membrane. I just have a mistake that no membrane. Actually, its role is that conformation is changed when ethylene binds with the receptor. The conformation of change and conformation change. That's why the CTR1 this cannot remain with this kinase, okay, or becomes activated. That means it remains bind with the domain, so it becomes activated. But when it further no longer binds with the domain, receptor's domain, that means it becomes inactivated. Clear? So what is the role of this CTR1 and the ethylene and the histidine kinase domain is clear now. I think so. So this is the ethylene signaling pathway, and how much you understood this topic, you can comment in my comment section. Okay. So please support me for the growth of this channel and continue with my channel. Thank you.